our last MVZ lunch of the semester. I have just a few quick announcements, and then I'll turn it over to Eileen to introduce Shannon. Tomorrow, the IB seminar is Alex Ophir from Cornell University. The title is The Cognitive Ecology of Early Life Social Experiences and Reproductive Decision Making. That's in VLSB at 12:30 in person. Friday, the herbarium talk at 12:30 is Forrest Freund, a grad student in the Rothfels lab. And this is a finishing talk, and I don't have the title for that. Essay brunch Friday at 10 is Sarah Harajan, a student in the Boots lab. And she will be speaking on DWV variants among island bee populations. I'm not sure what DWV is, but I'm guessing it's something virus. Okay, and I want to remind everyone that the MVZ symposium is next week, so no MVZ lunch on Wednesday. And I just noticed on the MVZ website, it says that the symposium is on Wednesday. That's not correct. I hope everybody knows the symposium is on Monday, May 2nd, and will be in the Brazilian room. So please pay attention to the announcements. Carly will send out reminders. And I'm delighted to see that just about everybody signed up to, all the students and postdocs have signed up to speak. Okay, that's it. That's all I have. Eileen, take it away.
Okay, well, hello from the Cono Sur. It's my great pleasure to introduce today's speaker, Shannon O'Brien. Today is Shannon's finishing talk, and so it's a chance for all of us to get together and to hear about her work and to help her celebrate her accomplishments at Berkeley. That Shannon's from the Bakersfield area, and I understand there's some family and friends on from that area right now on this call. So she's a native Californian, but she left the state to complete her BS degree at the University of New Mexico, where she, if I remember correctly, initially explored degrees in psychology and anthropology. But partway through her time there, she ran into the Museum of Southwestern Biology, and in particular, the mammologist there, Joe Cook. And it was her connection with Joe that convinced her to pursue a graduate degree in evolutionary biology. So Shannon arrived in Berkeley in August 2016, and she's been on a stellar trajectory ever since. That she came in as a Chancellor's Fellow and subsequently was awarded a Ford Foundation predoctoral fellowship. And while at Berkeley, she's published 6 peer-reviewed papers and has 2 more that'll be submitted in the next couple of months. In addition to her research, Shannon is a dedicated teacher who's really gone above and beyond by working with the Center for Teaching and Learning on campus, both to improve her own teaching skills, but also to help develop course content. And this was particularly evident during the enforced transition to remote instruction in 2020, when Shannon's efforts were really critical in helping transition the animal behavior course that George Bentley, Damian Elias, and I teach to an online format. So Shannon's a dedicated teacher who's really invested a lot in that aspect of her career. Throughout her time in graduate school, Shannon has also participated in multiple outreach programs, most aimed at promoting the role of young women in STEM disciplines. This includes several years of work, I should have advanced the slides, several years of work with the Expanding Your Horizons group, during which Shannon helped to design and implement workshops that have been attended by over 400 GK through 12 students. Shannon's also worked with the Graduate Women's Project and the Berkeley Science Review, both of which have provided opportunities for her to share her experiences with others in a written format. And these efforts have significantly shaped Shannon's future career trajectory. I'll say a little bit about that in a couple minutes. With regard to her dissertation research, Shannon has completed a particularly integrative project that explores patterns of social behavior in highland tuco-tucos. She's going to tell you all about this in a couple of minutes, so I don't want to drop any spoilers here, but I did want to emphasize how broad and integrative her project has been. So for her doctoral research, Shannon conducted fieldwork in the Puna of extreme northern Argentina. And due to both the elevation and the habitat, this is a pretty demanding place to work. But despite these challenges, including just simply breathing at times, Shannon successfully obtained critical behavioral data from her study animals over the course of several field trips. Now, based on these data and the associated samples, Shannon went on to develop expertise with social network analyses, with assays of glucocorticoid hormones, and with analyses of gut microbial diversity, all aimed at understanding the causes and consequences of variation in social relationships among her study animals. Now, this is a pretty impressive suite of tools and skills to bring to behavioral research, and Shannon deserves a lot of credit for pursuing such a diverse project. Now, just to wrap up so the introduction doesn't turn into the seminar, I'd like to offer a couple more personal observations that every mentor-mentee relationship is different. As a mentor, I hope that I contribute something to the careers of the graduate students that I work with. At the same time, I know that I learn something from each student that I work with. Now, one thing that I learned from Shannon is how to always look great in the field. As soon as I'd step out on the field site in the morning, I'd immediately be covered by dirt, and hair dye, and all kinds of other things. And Shannon would work right beside me all day long. Yet at the end of the day, her clothing was still spotless. She looked like she just, stepped out of, I don't know, I don't know. She looked perfect, right? You never know. She'd just spent 12 hours chasing rodents under pretty inhospitable conditions. I'm still trying to figure out exactly how she does that, but I did pick up a few tips along the way. More seriously, working with Shannon has taught me a lot about taking risks, trying new things, and yet remaining true to yourself. And as I hope is already clear, I think that Shannon's done an outstanding job with her dissertation. As part of that experience, she's realized that a career in academia is not really her best path. Instead, Shannon has discovered she is passionate about science writing and about using her education to help inform others. She's also a skilled graphic artist. A couple of her examples of her work are shown here in this slide. For example, she designed the logo for the 150 Years of Women at Cal program. But collectively, Shannon is realizing she wants to pursue a direction more related to communication of science and science education. And given those interests, I'm really pleased, I hope this isn't a spoiler alert, Shannon, but to let everyone know that just last week, she was hired full-time as a science writer for Galley, an online educational resource based in Europe. And I'm sure Shannon will be happy to tell you more about that if anyone's interested. But I'll stop there. And with that, I'll ask you to join me in welcoming science writer and soon-to-be doctor, Shannon O'Brien. Thanks, Shannon.
Thank you, Eileen. That was very touching. Let me pull up my screen one moment. Always takes a bunch of clicks. Okay. And here, I just learned how to do this type of screen sharing this morning. So here we go. Okay, everyone can see it? You could only see my slides, right? Okay, perfect. Well, thank you again. And hello, everyone, for being here. I really appreciate what you said, Eileen. That was really touching. Throughout graduate school, I always wondered what it is you would say during my finishing talk. So it's exciting to finally be here to hear it. So thank you all again. And today I'll be telling you about what radio collars, hair dye, and poop have taught us about highland tuco-tucos. But before we dive into our research, I want to give a couple of thank yous to some important players in this process. The first being Dr. Joe Cook, who Eileen mentioned. He was my sort of unofficial undergraduate advisor at the University of New Mexico, and he is the one who introduced me to biology and gave me my first research experience. He's the one that encouraged me to pursue graduate school, and he has been a constant source of support ever since. And then next up, of course, I would like to thank Eileen, my advisor, for accepting me into her lab and guiding me through this whole process. I know that this particular project is really near to her heart, and so I'm really thankful and grateful that she trusted me with it and allowed me to make it my own. Then finally, I would like to thank the tucos, Before I came to Berkeley, I admit I hardly knew what a tuco was, but they quickly grew on me, and now they are by far one of my favorite animals. So why tuco-tucos? And I don't mean why are they one of my favorite animals? I mean, why study them? And other than the fact that, of course, Eileen specializes in tucos, which helps a lot, it's because I'm interested in social behavior. So from the start of graduate school, I knew that I wanted to study something about animal sociality. And so thanks in huge part to Eileen's work, tuco-tucos have become an excellent comparative system for understanding sociality. So of the tucos that have been studied, the majority appear to be solitary, meaning that they live and nest alone in their own underground burrow. And that's expressed here on the left side of the figure, where you can imagine that each oval is an animal's home range, and each dot is that animal's nest site. And none of those ovals or home ranges overlap, which is why they are considered solitary. And then listed on the left is a few of the tuco-tuco species that follow this pattern of space use. Now, when we compare that with C. sociabilis on the right side, this is the colonial tuco-tuco, you can see from the figure that the individual's home ranges overlap extensively, and that within a group, there's a single nest site that individuals share. And this tells us that this species is social. But only 6 species are shown here, and there are over 60 known species of tuco-tucos. So what is happening with the rest of them? And that is where my dissertation research begins. I chose to focus my research on one of the many undescribed species of tuco-tucos to see where they fit into this spectrum of sociality. So my research focuses on the highland tuco-tuco, as Eileen mentioned. This is Ctenomys opimus. And the population I study is located within a national park in northwestern Argentina called Laguna de los Pozuelos, or just Pozuelos, as I'll refer to it throughout the talk. And this picture here gives you a good sense of what Pozuelos looks like. So on one side of the fence is the national parklands where we do our work with the tucos. And on the opposite side is private property with beautiful mountains and livestock in the background. It's really not a bad place to work. So for my dissertation, I covered 4 main topics. For my first chapter, I sought to describe highland tuco-tuco social structure. For chapter 2, I describe how their social structure changes over time. Next, I examined the relationship between social structure and glucocorticoids or stress hormones in this population. And then finally, for my 4th chapter, I examined the relationship between social structure and the gut microbiome. So today I'll touch on the main findings from each of these chapters, but of course we'll begin with chapter 1, which again is about highland tuco-tuco social structure. So prior to our work, there was no real quantitative data about the social structure of highland tuco-tucos. An initial observation of a Peruvian population in the late 50s suggested that highland tuco-tucos might be solitary. In fact, for a really long time, the majority of tuco species were assumed to be solitary, and it's only in recent decades that we're beginning to quantitatively describe their social behavior. So all that said, a colleague of Eileen's observed the population at Pozuelos and based on the way they were behaving, thought that they might be social. And so Eileen was called out to investigate. So in 2009, Eileen went to Pozuelos and started collecting data on the tucos to learn more about their social behavior. And this involved capturing the tucos and giving all the adults a radio collar to track their movements underground and some new hair color to help track them when they're above ground. We also collected info on their sex, reproductive status, and weight. And all the individuals captured were given a pit tag or a microchip to permanently identify them. So spatial data was collected from all the adults on the population in about a 1.5 hectare area using both radiotelemetry and visual scan sampling. But before collection began, the field site was organized in an 8 by 8 meter grid using a Cartesian coordinate system. And this essentially just means that there was a fixed corner of the field site, and then from that point extending in both directions, numbered flags were placed at 8 meter intervals to form a grid. And this grid is used to help identify where the tucos are when they're above ground. So that way we can observe them, but still maintain a safe distance so we don't disturb them. So during the collection period, adult spatial locations were recorded hourly during daylight hours. Additionally, spatial locations were recorded during 5 full days and nights, or 120 consecutive hours, to give us insight about how their behavior may change between daytime and nighttime. So after collecting hundreds of spatial data points from the adults on our field site, we're then able to generate home range maps that give us an idea of which individuals may be hanging out together. So this figure here uses 95% minimum convex polygons for all the individuals that were tracked in 2009. So 95% MCPs exclude the 5% most extreme data points for all individuals. So in other words, if individuals have like random forays away from their core home range, rather than overestimating the size of their home range, we're able to calculate a more conservative estimate since we've excluded those 5% of extreme data points. So this figure has a lot going on, but all you really need to know is that each polygon represents a single individual's home range. And when you look at the polygons, you can see how some individuals overlap with lots of other individuals, while others overlap hardly at all. And then from those data, we're able to calculate an overlap matrix, and that tells us to what degree each individual overlaps with each other individual. And I have an example here on the left with some made up data. So in this case, A, B, and C, and D, these are the animals. They are represented along the top and left side of the matrix. And the yellow area represents each individual relative to itself. So of course, overlap is 1 or 100%. So the numbers in the matrix represent the percentage of home range overlap between two animals. 0 means those individuals don't overlap at all, and numbers can range anywhere between 0 and 1. And these matrices can then be used to perform social network analysis, which provides us with more detailed information about how well individuals are connected within a network. It looks a little something like this. So here we have the daytime and nighttime telemetry data for the individuals that were tracked on our field site in 2009. Here, the gray circles represent males and the white circles represent females. And you'll notice that groups vary in size and contain a mixture of adult males and females. And when you look at the individual IDs within each circle, you'll see that some groups are stable during the daytime and nighttime, meaning the same individuals that overlap during the day are also overlapping during the night. And I've circled those groups here. You'll also see that the other daytime groups splinter into smaller groups at night, meaning that even though these individuals overlap during the day, they do not all nest together at night. So from this, we can already tell that groups are dynamic in terms of their size, their sex composition, as well as temporally. So in addition to examining group dynamics during daytime and nighttime, for a subset of animals that had enough visual data, we were able to compare above ground versus below ground groups. And remember, above ground individuals were tracked on their hair dye colors, whereas below ground, we tracked them using the radio telemetry. And we found, thankfully, there were no differences between these contexts. And this is really great, and it tells us two things. First off, it tells us that the tucos are pretty consistent in who they are hanging out with above ground and below ground. But secondly, it tells us that there's no significant differences between our two methodologies. In other words, their social structure pretty much looks the same whether we track them with visual scan sampling or radio telemetry. So now, when we relate our social network data back to our home range polygons, it really highlights the diversity in this population's social structure. So in this figure here, each colored polygon represents an individual's daytime home range, and the outlined black polygons show us their nighttime home range, so how they move after the sun goes down, basically. And then the black dot represents the nest site where individuals are sleeping. So here you'll see that individuals overlap extensively during the daytime and nighttime, and they're sharing a single nest site. And if you go back to that spectrum of sociality figure, this looks just like the pattern we observed in C. sociabilis, that social tuco-tuco. But the story does not end there. When we look at another group from the Basuelos population, something different emerges. So here we have a group that overlaps during the daytime, but at night, only 2 of the individuals, O and Y, share a nest, while the other 2 sleep alone. And then here we have another group from that population. Again, we have individuals that overlap during the daytime, those colored polygons, but they split into smaller units at night, either sleeping in a pair or alone. Except for this male Q, he did something really interesting. Some nights he slept with females T and U, and other nights he slept by himself. So really dynamic stuff is going on in this population. So from our data, we've determined that highland tuco-tucos are complicated. We found that groups range in size and can consist of multiple adult males and females, and that groups differ between daytime and nighttime, but are consistent above ground versus below ground. So if we relate that back to that spectrum figure again, it seems safe to say that highland tuco-tucos are social, but the groups are not as tight-knit as they are for C. sociabilis. So it seems like the highland tuco-tuco should fall somewhere in the middle of the spectrum. And then this leads us to my second chapter. So now that we've observed that this population is dynamic, we wanted to know how these patterns might change over time. And the reason that is because you cannot determine a system's social structure from only one field season. You need multiple years of data to confirm patterns you see are true to the system and not mere artifacts. So in other words, was that variability we saw in chapter 1 a stochastic event, or does this population consistently have that type of variation in social structure? So to answer this question, we went back to that same population and we tracked individuals across 5 field seasons. So same as before, we caught all the individuals within that same gridded area, but this time we focused only on visual data. So since our analyses from that first chapter show that there weren't significant differences between those two methodologies, radio telemetry versus visual scan sampling, we decided to go with just the visual data because it's a lot cheaper and easier to use, and it's a lot more convenient than radio telemetry. Also, without being limited by the number of radio collars we could afford, we were able to expand our tracking to not only include adults, but also subadults. So again, we did 5 field seasons, we used visual data, we looked at adults and subadults, but otherwise everything was the same as before in terms of methodology. So this figure here represents the total number of individuals that were captured each consecutive year from 2011 to 2014. And it shows the proportion of those individuals that were recaptures from previous years versus new captures that had never been caught before. So since this figure was created to show the number of recaptures, 2010 is not shown here because that was our first year of data collection for this study. So hence there were no recaptures. So here, pink represents recaptured adults, purple represents recaptured subadults, and blue represents individuals that were captured for the first time. So in 2011 and 2012, about 1/3 of individuals were recaptures. In 2013, there was a single recapture, and in 2014, about 15% of individuals were recaptures. So some quick important numbers for chapter 2. During the entirety of this study, 184 individuals were tracked. And of those, 27 individuals were subadults the first time they were captured. And then across the whole 5 years of this study, there were only 31 individuals recaptured. And none of those individuals were captured for more than 3 years in a row, which gives us a sense of their lifespan. So from these numbers, you can see that recaptures aren't super common in this population, but since we're interested in how social structure changes over time, the recaptured individuals are really the star of this study because they allow us to examine how consistent they are year to year in terms of their space use and their social structure. So here we have home range maps for all 31 of those recaptured individuals. So for these maps, each color represents the home ranges within a given year. And each mini map shows the home range for a single individual in each of the years that they were captured. So the main takeaway here is to see that some individuals stay in roughly the same place year to year. So those are the individuals with lots of overlap between their home range polygons. And other individuals have a distinct home range each year. So in other words, rather than staying in the same place, they move to a new location each year. So to bring this into further perspective, here we have 4 females, each of whom was captured during 3 consecutive years, 2010, 2011, and 2012. And you'll see that the female on the far left has a distinct home range each year, whereas the female on the far right has a home range in roughly the same location each year. And then the 2 females in between represent variations on those extremes. So again, these patterns highlight the diversity of behaviors that occur within this population. So with individual space use varying across years, it of course begs the question whether social groups are consistent across years. So right off the bat, we can go back to those initial stats I shared. So 184 individuals were tracked during this study. 31 of those were recaptures. So that leaves 153 new individuals coming in and out of the population across those 5 years. So of course, that's not really a great indicator of social stability. But of course, we did not leave it at that. We did a lot of statistics and so on, but I won't bore you with all of the details. I'm just going to tell you the highlights that we found. So we found that there were 6 individuals that appeared to be solitary, meaning they did not overlap with other individuals at all, but none of those individuals remained solitary for more than a year. Next, we found that most recaptured individuals lived in larger social groups over time, potentially suggesting the ability for individuals to better socially integrate as they age. Lastly, recaptured groups were not stable, meaning that recaptured individuals that were together in one year were not always together in subsequent years. So for example, you may have individuals A and B in a group in year 1, and then in year 2, they're both still on the field site, but now they're residing with different individuals. So I hope all of this together has convinced you that sociality is very dynamic in this population. And also an important thing to point out is that the variation we observed in this population is not due to differences between individuals. In other words, we didn't observe individuals that were consistently solitary or consistently social. Rather, everyone was doing different things. Everyone was dynamic. So to summarize the results from my second chapter, a dynamic social structure was observed across all years of the study, which tells us that this is a consistent feature of this population. We also saw that recaptures are not super common in this population, and that recaptured individuals are not very consistent in their behaviors. They may stay in one place or they may move locations between years. And then this leads to my next point, which is that groups are not stable across years. So if we bring it back to that spectrum figure once more, we can confirm from our data that highland tuco-tucos belong in the middle of the spectrum. They are social, but not to the same degree as C. sociabilis, because remember, we did observe some individuals that were solitary within the population. So now that we know that they belong somewhere in the middle of the spectrum, the next question becomes why? Why are the animals doing this? So now there are a host of variables that could be explored here, but we decided to first start with their physiology. So this brings us to my third chapter, where we were interested to see whether the variation in social structure observed in this population, if it correlates with variation in glucocorticoids or stress hormones as they were commonly referred to. And the reason we were interested in this is because past research done in our lab by Dr. Julie Woodruff showed that in Unearthing Sociality, that other social tuco-tuco, that there are significant differences in glucocorticoid concentrations between females that breed in their natal group relative to those that disperse and breed alone in their first year. And she found that individuals that bred alone had significantly higher corticosterone concentrations than those that bred in their natal group. And this pattern held both in the field and in the lab population at Berkeley. So we wondered if a similar pattern would be observed in our population of highland tuco-tucos. Would individuals that lived alone or in small social groups have higher GCs than those that lived in larger groups? In layman's terms, does sociality influence stress hormones in this population? So to examine this in our population, we too wanted to use a free living field population as well as a lab population. But since there are no captive populations of highland tuco-tucos, we made our own by capturing individuals that were off-site from our target study population. And we kept them in captivity for a few weeks so that we could conduct our experiments. So you can see from this little video that the tucos took pretty well to captivity, and they were very happy to eat the corn and grass that we fed them. So to examine glucocorticoids in the field population, we didn't have to do that much extra work in terms of our field methods. We just needed to collect feces from individuals when we first captured them, prior to monitoring their space use and sociality like we did for the previous chapters. So thankfully, tucos tend to poop when they're in their handling bags. So we just put those fecal pellets into a tube and froze them in liquid nitrogen. And the reason we chose to use feces as opposed to blood is because it's a relatively non-invasive and easy to collect measure, especially when we're out in the field. And it provides us with a general baseline measure of their glucocorticoid levels. For the lab population, things were more complex. So for our study, we kept 12 adults in captivity, 6 males and 6 females. Half of the individuals were housed alone. So we had 3 males and 3 females housed alone, and the other half were housed in male-female pairs. And these pairs were chosen based on field observations that indicated that the male and the female were likely from the same social group. They were in very close proximity, and they were observed using the same burrow entrances. So we had a pretty good sense that they were familiar with each other and would be comfortable being paired together. So these paired individuals were put into a cage that was separated by a mesh screen. So this allowed them to see each other, cuddle, and communicate, but they were still separate so that we could easily collect their poop. And all these animals were in captivity for minimally 1 week. This would allow them time to acclimate before we gave them any treatments. So once that acclimation period ended, we collected feces from them every 6 hours for a period of 48 hours. After those 48 hours, individuals were given an intramuscular injection of either adrenocorticotropic hormone, or ACTH, or saline for a control. So for our study, 8 individuals received ACTH, and 4 received saline. And feces collection continued every 6 hours after treatment up until 72 hours post-treatment. So for those of you who are unfamiliar, ACTH is a hormone that leads to the secretion of glucocorticoids from the adrenal gland. So ACTH is frequently used in studies to create a peak stress response in individuals to validate that this stress response can in fact be measured in their feces. So this is a really necessary validation experiment when you want to study fecal glucocorticoids in a new study population. So part of our motivation for holding animals in captivity was to do this ACTH validation study to ensure that we could in fact measure glucocorticoids in their feces. But we also use this challenge in combination with these different housing treatments to see if solitary or socially housed individuals responded differently to the treatment. So once fecal samples were back in Berkeley, I had the pleasure of cooking tuco poop to dry it out. And then I processed the samples with ethanol and performed extractions using a commercially available corticosterone lytic kit. So now for our results, I will start with the lab population. At capture, there were no differences in glucocorticoid concentrations between any of the individuals that we held in captivity. So that's good. That's what you want to see. You want to see that all individuals are starting from the same place. Then around the 12-hour mark, individuals that were treated with ACTH, that's the solid black line here, they reached peak glucocorticoid levels. And then that began to return to pre-treatment levels as time went on. In contrast, the control individuals that were treated with saline, represented here by the dashed line, they stayed relatively stable over time. So this is good. This tells us that the ACTH challenge worked and that individuals treated with ACTH responded, meaning we have validation that we can measure glucocorticoids in the feces from this population. Excellent. So now we can get into the more interesting stuff, like the different housing treatments. So this figure shows both the ACTH versus saline treatment, as well as housing treatment. The dashed lines represent all the saline or control individuals. And you can see that there were no differences in glucocorticoid concentrations between housing treatments for those individuals that were treated with saline. However, when you look at the solid lines, those are the ACTH treated individuals, you see something different. So there were no differences in their concentrations except during the 48 to 72 hour time period. So you see highlighted on the right side, the ACTH treated individuals that were housed with the partner, that's the black line, they had significantly lower glucocorticoid levels compared to the individuals that were housed alone, that's the pink solid line. So this is a really small sample size, but it leaves an intriguing place for future study. Like perhaps sociality helps mediate stress in this population. Maybe individuals are able to return to baseline or recover from stressors more quickly when they're in a social environment. So now we're going to jump to another figure. Again, we're looking at ACTH versus saline treatment, but this time in relation to sex. So same as before, the dashed lines represent all the saline or control responses, and you can see that there were no differences in concentrations between the sexes for those that were treated with saline. However, when you look at the solid lines, those are the individuals treated with ACTH, you can see that there was a significant difference between males and females. We found that males had significantly higher glucocorticoid concentrations than the females. So let's keep all of that in mind and compare it to the field population real quick. So remember, we collected feces from the tucos when we captured them in the field, and then we tracked their space use so that we could do social network analysis. So here are the social networks for both years of the study. In this case, the nodes, or the individuals, are sized proportional to their glucocorticoid concentrations. So larger nodes represent higher glucocorticoids. And perhaps you can already tell, there doesn't seem to be that much of a pattern. Glucocorticoid concentrations were really all over the map in our field population. And in fact, we didn't find any statistical relationships between GCs and our social network metrics. So despite our lab housing experiments, which suggested that maybe sociality helps mediate stress, we did not find any support for differences in sociality affecting glucocorticoids in the field. However, remember our lab population, we did find that males had higher GCs than females, and we also found the same in our field population. So now we have support from both the field population and the lab population that there are sex differences in glucocorticoid concentrations in this population. So why might that be the case? Like, what is going on with the males? There could be a few things going on, like maybe males and females deal with stressors differently in this population, or may have to do with intersexual differences in reproductive behavior. So something important to mention here is that these experiments occur during the spring breeding season for this population. So that may be driving the higher GCs in the males. Similar studies in other rodent species have shown sex differences in glucocorticoid concentrations for some species. In other species, they've shown no sex differences at all. In some species, males have higher GCs, other studies, the females do. So the literature is a bit of a mess. There's no real clear pattern, but it's likely that environmental and phenotypic parameters are playing a role. So to summarize the results from my third chapter, again, highland tuco-tucos are complicated. That's the theme of this talk. And variation in sociality does not seem to affect glucocorticoids in this population. At least it wasn't observable in the field. And there's a possibility that sociality helps mediate stress, but of course we would need to do more lab experiments to confirm that. So rather, our big finding for this chapter was that sex was the best predictor of GC variation, with males having much higher corticosterone concentrations in both the lab and the field population. So we didn't find what we were expecting, but we had one more thing we wanted to explore, one more potential variable that could possibly help explain why the highland tuco-tucos have this dynamic social structure. And that brings me to my final chapter. For this chapter, we were inspired by all these papers coming out that suggest a relationship between sociality and gut microbiome diversity. So a little quick review of the microbiome literature. Basically, sociality is thought to increase gut microbiome diversity. The thought being that as you come into contact with conspecifics, you may exchange microbes, which in turn makes your gut microbiome more diverse. Similarly, sociality is also thought to increase gut microbiome similarity between individuals. In other words, if two individuals spend a lot of time together, they're likely to have more similar gut microbiome profiles relative to individuals that they don't spend any time with. So the majority of research being done on this theme has been in laboratory mice or in wild primate populations. So either populations under tight laboratory control or species like primates that maintain really distinct social groups. So we wanted to know how this relationship, that is the relationship between sociality and the gut microbiome, might play out in the highland tuco-tuco population. So same as we did for the feces collected for chapter 3, we kept some of those feces collected at first capture so we could study the gut microbiome of these individuals. So in 2017, feces were reserved for this purpose. They're preserved at an ambient temperature in RNA later. In 2018, feces were immediately frozen in liquid nitrogen without RNA later. And this might be raising red flags for you. I'm sorry, I just forgot to pack RNA later during my second field season. So we work with what we have and we move on. So once feces arrived at Berkeley, they were stored in a -80 degree freezer. Next, I extracted the DNA from the fecal samples using a Mobile DNeasy Power Soil Kit, and then sent the DNA to Microbiome Insights in Canada, where I let the experts handle the rest. So for those interested in the details, Microbiome Insights sequenced the V4 region of the 16S rRNA gene on the Illumina MySeq platform, and then quality filtered the samples and clustered them into 97% operational taxonomic units, or OTUs, and high quality reads were classified using the Green Genes database. So for this study, we were able to collect feces from 15 adults in 2017 and 14 adults in 2018. There were 4 individuals in 2018 that were recaptures from the previous year, but we considered them independent for our statistical analyses. So we found that Bacteroidetes and Firmicutes, if I'm pronouncing those correctly, were the dominant phyla, which is typical in mammals. However, we found that the relative abundances of these phyla differed between years. So in 2017, relative abundances of Firmicutes were higher, and the opposite was true in 2018, where the relative abundances of Bacteroidetes were higher. So now when we look at the data from both years on a PCOA plot, you can see that the samples cluster quite strongly by year. So here the dark blue are the samples from 2017 and the light blue are the samples from 2018. So this brings us back to those different preservation methods each year. That's probably driving this difference. So when we did our data analysis, we considered each year separately. So now to look at the relationship of sociality and the gut microbiome, we looked at 2 main metrics of gut microbiome diversity, alpha diversity and beta diversity, but I will start here with alpha. So alpha diversity is the diversity of the microbiome within an individual. So if you were to look inside one individual's gut, what is going on in there? Is it really diverse or is it not? And this is measured with the number using the Shannon Diversity Index. So remember, sociality is thought to increase gut microbiome diversity. And this happens via a process known as horizontal transmission. So this is the transfer of microbes from one host to another. And it can occur from all sorts of social interactions like huddling and grooming, or even like fighting and mating. So we use our social network metrics that we calculate for our individuals in the population as predictor variables in linear mixed effect models to see which, if any, predicted alpha diversity. And we found that while social network metrics were significantly correlated with alpha diversity, the models were not strongly predictive. So social network metrics do not seem to be predictive of alpha diversity within this population. So now on to our other metric of interest, beta diversity. Now, this is the similarity of gut microbiome compositions between individuals. So if you look at the guts of different individuals, how similar do they look? And this is measured using Bray-Curtis dissimilarities. So like I mentioned before, sociality is also thought to influence beta diversity, also via horizontal transmission. The thought being that as individuals come into contact and share microbes, their gut microbiomes may become more similar, whereas individuals that are separate from each other or don't interact are likely to have more distinctive, less similar gut microbiomes. So this is analyzed by examining how samples cluster together. So similar samples should cluster. So if sociality plays a role in beta diversity, then we should see that samples cluster together by social group. But unfortunately, samples did not cluster by social group at all. Here the data are shown separately for each year, 2017 and 2018, and each color, it represents a single group, and they're just all over the map. Like the colors are not grouping together. There's no sense of clustering. So again, samples did not cluster by social group. We had thought that maybe we weren't seeing a strong social signal because social relationships are not tight-knit in this population. So typically, species that do show a strong social signal are those that have really close groups that are distinct from one another. But you remember from previous chapters how dynamic these tucos are, and that their social groups are by no means stable. So instead of examining beta diversity in relation to social groups, we wanted to look at it in relation to home range overlap. So now remember, these tucos move primarily underground through a network of tunnels. And so when home ranges overlap, it's highly likely that these animals are using the same tunnel networks, eating in the same tunnels, pooping in the same tunnels, and so on. So that leaves a big potential opportunity for individuals to share microbes, even if those two individuals never actually come into contact directly or are not in the same social group. So we compared our home range overlap matrix to our beta diversity scores, and we did find a significant relationship. So what could this mean? Well, something to bear in mind is that tuco-tucos are tooth diggers, at least in part. So as they're using these tunnels and digging new ones, it seems plausible that they may consume the feces of other individuals along the way, even individuals outside of their social group that they otherwise would not come in contact with. So now coprophagy, or eating poop, isn't a regular part of the tuco diet like it is in some other species. But even so, we have observed it occasionally in the lab. So it stands within reason that this is happening in the field, either directly through coprophagy or even indirectly during tooth digging. So this is something exciting to think about. Could tooth digging be a mode of like contactless horizontal transmission? What if individuals can transfer gut microbes with conspecifics that they never come into contact with? So to summarize the results from this last chapter, hopefully you've guessed it by now, highland tuco-tucos are complicated. Contact with individuals outside of primary social groups may be a stronger mode of horizontal transmission in this socially flexible population. And this could be occurring via tooth digging, coprophagy, or shared use of underground tunnels. Now, for those big microbiome people in the audience, I'm sure you're thinking like, what about kinship? What about vertical transmission between related individuals? And that's a fair question. We are working on that. We have those data. We're currently working on microsatellite analyses, so those data are forthcoming. So now to quickly wrap up the main takeaways from the whole dissertation. In chapter 1, we learned that highland tuco-tucos are social. Groups range in size and can consist of multiple males and females. Groups may splinter off at night, but are stable above ground versus below ground. From chapter 2, we were able to confirm that a dynamic social structure is a consistent feature of this population. And we learned that recaptures have low site fidelity and low group fidelity. So in other words, home ranges and group structure are not stable over time. And this tells us that the dynamic sociality observed in our population is not due to consistent differences occurring between individuals, but rather everyone is acting dynamically. Next, from chapter 3, we learned that sociality does not appear to impact glucocorticoids in this population, but sex does. Males have higher corticosterone concentrations than females. And then from chapter 4, we learned that contact outside of primary social groups may prove to be more important than contact within social groups for the transmission of microbes in this population. So what does this all tell us bigger picture? Where does this leave us now? Well, to start, it confirms that there are intermediate forms of sociality in tuco-tucos. And this is exciting because it will help to continue to build that comparative framework within Tanomys. It also tells us that not all forms of sociality are super predictable. And this leaves us with a lot of room for exploring dynamic forms of sociality. Sociality may help mediate response to stressors, acting as a sort of buffer to help individuals recover from stressful situations more quickly. And in socially flexible species, contact outside of primary groups may prove a more predictive measure of gut microbiome diversity than considering contact within social groups alone. And that brings up the related point that tooth digging may be a mode of contactless horizontal transmission, which opens up exciting avenues for future research. So in short, to wrap all that up, highland tuco-tucos are complicated. But before I take any questions, in proper finishing talk tradition, I want to thank all of my people. So for the sake of time, I will keep this brief, but for starters, I would love to thank my family. Even when they did not fully understand what I was doing, they supported me. And I am so grateful and really honored to be the first person in my family to earn a PhD. It means a lot to me. Excuse me, I didn't think I was gonna cry. And thank you to my fiance, Ignacio, or maybe I should thank Eileen for introducing us when I was interviewing to attend Berkeley. But jokes aside, he has supported me through so much throughout this process, so thank you. Thank you to my friends that helped keep me sane through all of this. They always kept my spirits up and helped me get through the tough times of a PhD. I, of course, have a big thank you to everyone in the MVZ, but especially thank you to Lacey Lab, past and present, for providing me a home away from home. And same for all of my office mates through the years. Thank you for giving me a safe space to talk and vent. Thank you to Lydia and Brian for all of your guidance in EGL. Thank you to Chris Conroy for helping get my samples from Argentina. Thank you to my cohort within the MVZ. We were like a really big cohort. And it always felt good knowing that there was a group of us going through all the stages together. Thank you to Michelle St. John for all of your help with my modeling questions. Thank you to Lelania and Carly for always knowing where everything is and knowing who to talk to get things done. I'm sure I'm probably forgetting someone, but this community as a whole has provided me with so much support. So thank you, everyone. Of course, none of this work would be possible without Eileen's guidance in the field. So thank you. Also, huge thank you to my field assistants over the years, Josefina, Juan, Christian, and Flor. Especially thank you to Christian for taking all the fabulous tuco pictures that you saw in this presentation. We found out on Monday that one of the tuco photos Christian took at my field site will be featured on the cover of Hormones and Behavior. So we're very, very happy about that. And thank you to Flor for all she did to get my samples out of Argentina. She handled a ridiculous amount of paperwork and finding dry ice and packaging and handled all of the logistics on the ground. So thank you a million times for making my dissertation possible. Thank you to George and Damien for serving on my committee and letting me work in your lab spaces. Thank you for being approachable and goofy and not letting me take myself too seriously. And also a huge thank you to Matina for teaching me how to do ELISAs. You made chapter 3 possible. And thank you for sharing your lab bench with me. Thank you to my dog Bunny, my favorite animal of them all. She got me through college and now she has gotten me through grad school and she means everything to me. And then lastly, I'd like to thank all of the sources that provided funding to me and my research over the years, especially thank you to the Ford Foundation and Berkeley Chancellor's Fellowships for giving me the gift of time. It can be really tough in graduate school to find a balance between research and teaching and all of our other commitments, and having those fellowships really allowed me to focus on my research and help me to make it what it is today. So thank you to them. Now, finally, here's a cute baby tuco to end on. Thank you all so much for being here on Zoom with me today. I really appreciate it. It means the world, and happy to take any questions.
Great, thank you, Shannon. Let's all either digitally or unmute for a second and clap. Big thanks for a great talk.
Hey.
Thank you.
I realize some folks may have to take off to go do other things, but I'm sure Shannon's happy to hang around for a little bit and answer questions, either suggest maybe raising your digital hand or putting a question in the chat box, and we'll field things that way. So, questions? Silence.
There's none, yeah.
Well, I have to leave in a second, but Shannon, I'm really excited for you.
Thank you.
You're going on to new things, and I really appreciated your talk. And I'm really happy you didn't fuck it up. You did really well, and yeah, I really want to see how well you do in the future. So congratulations.
Thank you so much.
All right. Other comments, questions? You blew them all away, Shannon. They're all processing
Great.
how weird tucos are. Okay.
Yeah, they're weird ones. That makes my life easy.
I had a question about the males.
Where are you? I can't see you.
I don't know where I am. Here, wave. Okay. I mean, you talked about this some, but I mean, are they fighting a lot more, or just traveling a lot? Or I mean, why would the males be so different than the females? Besides, you know, that seems really interesting to me. Like, why are they stressed out all the time? And
Yeah. That's a great question. Well, I think a big part of it is that we were studying these animals during their breeding season. So I think it would be really informative to go back out in the field during their non-breeding season, and maybe the males would be calmed down by then. But if not, yeah, that's a great question. Why? What's going on with them? They do maintain larger home ranges than the females. So maybe there is some sort of like competition going on between like the territories of males. But at the same time, there are social groups that contain multiple adult males. So they're weird. I don't know what they're doing. There's lots of potential research that can happen with this population.
Okay. Okay, thanks.
Anybody else?
Shannon, this is Michael. That was a great talk. You didn't say much about reproductive patterns. What do you know about are females mating with multiple males? Are males mating with multiple females? The overlap of home range is intriguing.
Yeah, thank you. That's a great question. And that is something we're working on. So we do have data, little toe clips from all the animals that have ever been tracked. And we're just working on figuring out the microsatellite analyses. So future, we will be able to tell you, like, are these animals in groups related or are they not? Who is producing these pups, who is not? We just don't have that information yet.
What's the litter size?
Well, in the lab, it can be like anywhere from 1 to about 5 or 6. Is that right, Eileen?
It is, although that's a different species.
Yeah,
I mean, Michael, it's a great question. And honestly, we don't know a lot yet. That the suggestion is, unlike most tuco-tucos, this population has 2 rounds of reproduction per year. The biggest one being November, December, January, when we were always in the field, but then a second peak in May. But to be honest, and I don't fully understand why, it's been super difficult to catch juveniles. Like, they just don't exist very commonly. And so it's a little bit of a mystery or a hole in this. Yeah. All right. Other questions?
Hey, Shannon, I have a question. Really cool to see how your dissertation worked out. So one of my questions is about the microbiomes stuff, whether you explored any other metrics other than Bray Curtis. So I've found that some of the other ways of measuring dissimilarity can really change how things group. And my second question is, do you happen to have samples where you have both the court measurements and the microbiome, or did you have to like basically prioritize those? Because it'd be interesting to look at what effects stress has on the microbiome and things like that.
Excellent question. So for the first part of your question, no, I have not explored other metrics yet. So I'd be interested to talk to you about that. For your second question, yes, we did look at the relationship between their court levels, and there was no relationship at all. When we did our modeling, the court measures were actually the least predictive of everything, like all the social metrics, group size. Court was the least predictive.
That's really interesting. Thanks.
Yeah.
Other comments, questions? Lots of folks are still here, but maybe they're all still pondering the mysteries.
Easiest defense ever.
Okay, well, folks can, Shannon, maybe it was in the pre-seminar conversation, she's working remotely right now as well, but I'm sure she'd be happy to answer questions by email if anyone wants to follow up and have questions occur to you. And Ammon, yeah, we'll probably follow up with you because that's the last piece that we've been trying to pull together in the dissertation.
Sure, anytime.
Thank you.
So thanks everybody for taking time to hear about Shannon's work, and yeah, see you all.